is get out of the way. Everybody should take photos of the board. This is a quick overview of some of the stuff that we're going to talk about. Now, what I want you to do in this lab today, I want you guys, and you're going to hear this a lot in this class, you've probably heard it already, because it takes so long to flip back and forth between the camera and the computer, I'm going to have you guys pull up the study guide on your phone so we won't do a lot of flipping back and forth. Because most of what I'm going to be doing is read off this and then take a source, one of those three, to find good pictures as I talk. Does that make sense? Mm-hmm. All right. So make sure you have this up on your phone. And what I'm going to do mostly is going through the things that it says right here that you need to know. This is very different than the lab that I was doing on skin. It had like a long list of terms at the end. This has a long list of objectives. So we need to be able to get through the objectives. What we're going to do is objective, then bounce to a table. Objective, bounce to a table. Insert pictures that help show things. Are you with me? You got the basic idea? All right, so let's start with the easy. Name the 4 primary tissue types in the human body. There are 4 tissue types. They are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Are you with me? Yep. All right, and I've already got details about all of those. So we're going to talk. Yeah, I came in early to write stuff on the board, so be ready. But we're going to talk about all this stuff. And as we go through the objectives, those of you who have your study guide up, your histology study guide, you'll notice that just like when we did the parts of the body, there's a list at the end. There's a table at the end, and it says what you need to know. Now, I'll make it really clear. This table 1 is 4 different epithelial tissues, okay? In fact, I'll go up to the objective number 2. Know the function and location of the 4 epithelial tissues listed in table 1. Now, if you look in your book, there are a lot more types of tissues than that. But, you've already learned this from the first test, what am I probably going to do the test questions on? The things in the study guide I'm supposed to teach you. So for epithelial tissue, those are the 4 I care about the most. I might do one of the others, but those are the 4 I care about the most. Does that make sense? The ones in the table. So where it says on table 1 that for each one of these 3 tissues, the simple squamous, simple cuboidal, pseudostratified columnar, and stratified squamous, that you need to know their function and location, those are the most important questions about the epithelial tissues. Does that make sense? All right, so we got that. Now let's talk a little bit about epithelial tissue. There are some things I want you to know about epithelial tissue. And I'm going to write something down that I'm sure I don't have on the board yet. Looking for a red marker. Here we go. I want red because we want to talk about blood. The short version is it doesn't have any. You with me? Epithelial tissue, I'm going to use the nerd words here, but it is avascular. That's a U, I promise. Avascular, which means without blood vessels. It's avascular and innervated. So now we'll say it in common speech instead of anatomy speech. It's got no blood vessels, but it's got some nerves. Good? No blood. Some nerves. Now when we say some nerves, I'm going to go for your lab book, and I'm going to show you a picture. We're going to use a picture on chapter 5. I'm actually going to use the picture from skin in chapter 6, even though this is about chapter 5. The reason is they have a real nice picture I said something nice about your lab book. Pretend to be shocked. It won't happen a lot, but they do have a really good picture here on the nervous system, on the skin. And what I'm zooming in to try and show you, I'm going to move it again so I'm out of the, we still have white space you can see. In the skin, we have these things called dermal papilla at the top of the dermis. Remember, papilla are like finger-like extensions that stick up? And they have nerves in them. A lot of them have nerves, not all of them. Some of them have blood vessels in them. But we also have nerves that go up into the epithelial tissue. So this is dermis, and once we cross up into the purple, we're epithelial. You can see we have nerve endings that go into the epithelial tissue, yes? So that's what I mean when I say innervated. Good? Most of the nerves may be down below in the dermis, but there are nerves up in the epithelium. None of these blood vessels are going to cross up. None of the blood vessels go into the epithelium. Good? All the things we're talking about in epithelium and skin is this top part here. All right? Anyway, but if you're taking notes, that is figure 6.1 on page 134. I'm putting my glasses. 124 in your lab book. Good? Clear? All right. I'm not seeing people write that down. That's okay. All right. So now, let's talk about the epithelial tissue. I gave you an example of innervated but avascular. You've now seen it with a picture of the skin. Let's talk about it. When we are talking about epithelial tissue, there are three things that we care about. And they're, by the way, they are the headings for tables 5.1, 5.2, 5.3 in your lab book. Okay? Table 5.1, I should write that in. So you know, these are tables 5.1, 5.2, and 5.3. Everybody good? And table 5.1, it talks about the layering. When we talk about tissue, it could have layers or not have layers. If we've got just one row of cells all attached to the same basement membrane, we're going to call it simple. You with me? If it's got more than that, we're going to call it stratified. So let's draw a little picture here. Say we've got some collagen fibers here forming a basement membrane, and above that we've got some cells. And we'll give them big round nuclei. We'll talk about that later. This would be a simple tissue because it's got one layer of cells. Do you agree? Now, let's give it another layer. It might line up the same, they might line up differently, but we've got another layer of cells. If you have more than one layer, you're going to describe that tissue as stratified. Are you with me? Like geologic strata, layers of the rock, we got layers of the tissue, it's stratified. All right, so simple or stratified, those are our two options that they talk about in table 5.1. The other thing we might talk about is the shape of the cells. If they're tall and thin and their walls are really close so the nucleus gets squished and looks kind of vertical, there's a picture of that in your book. Anyway, that's called a columnar cell. And there's a picture of that in your table 5.3. You see how those cells are tall like architectural columns and the nucleus is squished real narrow? The shape of the nucleus is real important when we're talking about cells and tissues, because sometimes, like in this photo, you can't get a nice focus to make out all the membranes, but you still know these membranes are really close together because, what was it doing? Don't do that to me. Because the nucleus is squished all tall and narrow. Does that make sense? If you have an oval nucleus, you have a columnar cell. Good? Good? All right. So, columnar cells, tall and thin, like that. Now, you might have cuboidal cells. When they say cuboidal, the cell is about the same height as width, And the nucleus, I'm going to draw this nucleus better. The nucleus is very, very round. So you have a very round nucleus. It's probably a cuboidal cell. And again, your textbook has some examples, has some good ones. I'm going to use this right here. Where we go? I'm in the wrong row. That's why I'm not getting it. All right, I'll move this up here. There we go. So these cells, would you agree? Very round nucleus, and kind of about the same height as width. Some of them, like this one right here that I'm working on, very much so. Some of them, of all the tissue types, the ones that I most like, use your imagination, is the cuboidal cells. Because this clearly is a little taller than is wide, but it's still a cuboidal cell. Are you with me? When you see a round nucleus, you should be thinking that is a cuboidal cell. Good? Now I'm going to show you another view. What you're looking at, I'd be like 99% sure, is a kidney tubule. I don't think they say it in this picture. Yeah, they don't say it, but they do say what they're used for, secretion and absorption. All right? I'm going to show you another picture on the next page, on page 95, I think it is. I shouldn't have taken my glasses off. Yeah, on page 95. This is also cuboidal cells, and it is the same thing. In the picture on page 93, you're looking at a kidney, even though they didn't label it, I guarantee you. You're looking at a kidney tubule with a cut down the tube. Good? In the picture on 95, you're looking at the same thing. You're looking at the kidney tubule, but now you're looking at the cut going across the tube. And that really shows what I'm talking about. The nice round nucleus, the nice round nucleus. The nice round nucleus. But let's talk about the cubeness. Would you agree this shape is not very cube, it's more trapezoid. This is more rectangular. This is more trapezoid. This one is not a proper cube, but it's a flat rectangle to the side. All of these are still cuboidal cells. Like I said, you have to use your imagination. This is simple cuboidal tissue. Simple, because we got one layer. The wall of the tube is one layer thick. Cuboidal, because this is all considered to be cuboidal, even if some of them are more trapezoid-like. Are you with me? Does it make sense? So when you hear something about cuboidal, this is a use your imagination to get the shape. All right, so we talked about that. The next one we're going to talk about is squamous. All right. And squamous is super easy. It's probably my favorite one. Yes, you're allowed to have favorite cell types. I'm going to let you guys pretend to be shocked. I have an empty Mountain Dew can. I wonder where that could come from. All right, you're all supposed to laugh because you know I drink Mountain Dew nonstop all the time. Nobody laugh. I'll pretend that you like my bad jokes. You do understand if you don't laugh at the jokes, I'll just try harder and it'll be worse for everyone. All right, so let's just work with this. So this can be squashed flat. Do we agree? That cell is squashed flat. The S in squamous, the SQU in squamous, the SQUA in squamous should remind you of the word squashed, because that cell is squashed flat. Very, very flat. All right? Now, let's go for some pictures. Are you with me? Nobody's like, woohoo! Okay, but we're going to try it anyway. We'll see if they have a picture I like. Well, they have some pictures, but I'm sure not liking them. This is stratified. We could go for simple. These are definitely squamous, but these in your textbook, your lab book on 98, I'm trying to focus to get better. And let's darken it up, see if that helps. No, let's go lighter. Does that help? Yeah, lighter helps a little. Would you agree this cell is super squash flat? This is its nucleus. That's a squash flat cell, yes? But that is a stratified squamous. Stratified means you got layers, yes? I'm going to show you better pictures. This is where I admit that your lab book is not your favorite lab book. Let's first go for the lecture book, because the lecture book in general is better than the lab book. I would rather show you things out of your lecture book than the old lab book, because like, well, what good is that to me? All right, so let's take a look. Here we go, here we go. There we are. So, simple squamous is a single layer thick. This is the basement membrane down here. And we have a single layer of cells on top, and they're very flat. There's the nucleus. Everyone good? That's a pretty drawing. This is the real thing. That's your lungs. You know why we wrap our lungs in ribs? Because they're really delicate. The air sacs are one cell thick. Here's an air sac, here's an air sac. That's what's between them. See how flat and thin they are? You can see the nucleus in there. Those are simple squamous. Now, you guys see this one right here? That's not an air sac. Anybody know what that is? That's a capillary. So the distance between this air sac and that blood, it's a little bigger. Yeah, I can even outline it. You can see it right here kind of. It's 2 cells thick. One simple squamous cell that makes the wall of the capillary, and one that makes the wall of the alveoli, the air sac of the lung. Does that make sense? Okay, so super flat. There's not much controlling the movement of oxygen into blood or carbon dioxide into air that you're going to breathe out. Because simple squamous, really thin, not much in the way, good for gas exchange, also good for filtration. The filtration membrane in your kidneys, the glomerulus is what they call it. You've got wall of the capillary, and you've got the wall of the Bowman's capsule, and you have the stuff flow right across. All the things that's going to become future urine. Does that make sense? Yeah, so thin stuff, good for filtration, good for exchange. I'm going to give you one other thing it's good for. We talked about some membranes before, 3 membranes that were super, super thin, super, super slippery. An S name for the group of them. Serous membranes, you remember? What was the one around the heart? Pericardium. What was the one around the lungs? Say it loud, I think you had it.
Pleural.
Pleural, yes, pleura. Okay. What was the one around the intestinal membranes? The abdominal organs, including the intestines. The peritoneum, yes? All of those are simple squamous too. Good? All the linings of blood vessels, lining of the heart, flat, slippery, smooth, those are simple squamous. Good? All right. So, would you agree we've talked about locations where there's simple squamous? And that was one of the things on the table. The other one was function. The function is easy gas exchange, no surface things that cause resistance. Good? All right. So simple squamous, really good at exchange, really good at filtration, stuff like that. And we've done one of the 4 epithelial tissues on the table now. But we talked about squamous. I'm now going to talk about one that is on the table. But it's a shape we haven't talked about yet. We've talked about columnar, we've talked about cuboidal, we've talked about squamous. Now we're going to talk about pseudostratified. Are you with me? So we're going to draw us the tissue. I'm going to draw a tissue up there. We're going to draw us the tissue down here. We've got some collagen fibers that form the basement membrane. And we're going to draw it tall. And you might say, oh, he's drawing columnar tissue. All right, so this, oh, it's columnar tissue. I'm going to divide it like that, and we'll still say it's columnar because it's taller than it is wide. Do you agree? But here's the thing. Columnar tissue, you'd be having this go on with the nuclei. Do we agree? Do you agree? Now watch something really weird, something really strange. This would be simple columnar. This would be stratified columnar. You agree? Here comes the weird stuff. We got the same basement member, we got the same apex. We're going to say we have some cells that are shaped like this, some that are shaped like this, some that are shaped like this. They don't even reach all the way to the apex. They're just like that. Leland, if this was not, like say someone's bad at a microscope and they can't focus it, could you imagine a situation where they think there are 2 rows of cells because they see 2 rows of nuclei?
Yeah.
Yeah. Okay. This is not stratified tissue, but it is pseudo-stratified. It's fake stratified. It looks kind of like it if you're not paying close attention, if you're not focused enough to see the membranes. How do we know this is actually a simple tissue? Because all the cells, even the ones that are barely attached, they're attached to the basement membrane. The ones that are broadly, strongly attached, they're attached to the basement membrane. The ones that don't even reach all the way up to the apex, they're attached to the membrane. They're all on the same membrane, so they're all the same layer. It's pseudo-stratified. Are you with me? Now, I'm going to change this a little bit to look a little narrower. We've still got pseudo-stratified. But, I'm going to do some other things. We'll talk about, we're coming back to this one. You with me? Now we're going to talk about the third specialization for epithelial cells. What is on the top of it? If it's got cilia on it that beat to cause things to move, think like the lining of the fallopian tube. We got an ovary, we got a uterus, we got to move an egg from here to there. We have the cilia making a current to wash the egg to the uterus. Does that make sense? Okay. So if you had cilia on top, there you go. That's one of the specializations. Say you had, because you do, say you had cilia on the top of this. I'm gonna draw in some cilia. Remember, cilia are the ones that beat like that. Yes, the pump. Okay. So I'm drawing cilia. I'm not going to draw them here. We'll talk in a minute why. All right. So you could have cilia. You could also have cells that have a thing on top we call microvilli. You saw some pictures before. They had up, down, up, down, up, down, up, down, instead of a flat membrane. Their membrane had lots of stuff on the top. That's to give extra surface area to absorb stuff. An example of that would be like kidney stuff. I'm looking at your textbook right now, and I'm going to find you one. Is that making me happy? Is that not making me happy? Come on, people. I'm about to go. Oh, well, we'll go with this drawing. I'll find a photo later. But this drawing is in your textbook. It is on page 162 for those of you taking notes. Do you see that the top of these cells have a bunch of microvilli, yes? Increased surface area for absorption. That's intestines to absorb the nutrients out of the food that's going by. Does that make sense? In fact, we got a special name for this thing of this whole layer of microvilli at the top. We call it the brush border. And in fact, when you do digestive in AP2, they'll talk about the fact that you even have little enzymes that are only found in that membrane. They're called brush border enzymes. They do some of the final parts of digestion before you absorb. Literally, nutrients going by like, oh no, we're going to break that protein further apart. We're going to break that sugar further apart. Does that make sense? Yeah. Brush border enzymes. Okay, so you've got that. Let's see if we can find a pretty photo. No, we're up to connective. I don't like it. I'm going to get you a good photo of the old lab book. I've been trying to avoid that. So We're going for it. So this is the old lab book's picture of simple squamous. Same thing, lung tissue. See how beautiful it is? See the little capillary? Here, this is that intestinal tissue. It's not zoomed in as much as the pretty pictures or the drawings, but this is that brush border made out of the microvilli for absorption. Good. It is simple columnar, but it's got this. It's got this light staining, but you see some granules in it. Yes. This is called a goblet cell. This is one of the specializations you can have. The goblet cell is insane. It is crazy. It is so hard to think about. A goblet cell is a gland that is only one cell. One cell all by itself is a gland. Do you know what that makes? It makes mucus. Well, let me say it another way. It makes a protein called mucin. And when mucin touches water, mucin becomes mucus. It changes shape and it absorbs the water, and now it's mucus. All right. So any layer that has goblet cells in it, they're making mucus. Now, there are different forms. There are forms that are real watery and they're used for lubrication. An example of that would be goblet cells in the intestine, like we're looking at there. Whenever they spill their mucin out, whenever they spill their mucin out, let's see if I can find the same picture again real fast. Probably not. I can find it real fast is the question. Yeah, we got it. When they spill their mucin out of the goblet cell here or out of the goblet cell here, and it hits water, I'm talking about intestinal goblet cells, they make a thin mucus. This lubrication to help the food go down the intestine. Does that make sense? Now, there are other tissues that have goblet cells. And what I drew over on the side here for the pseudostratified, that's one of them. Let me get my sheets of printer paper. Woo! I'm going to draw a goblet cell. You already saw photos just a minute ago of goblet cells. So light staining, but with some granules of mucin proteins. Yes? Good? This pseudostratified, ciliated tissue with goblet cells, that's the lining to your trachea. That's to keep bacteria and pollen from getting into your lungs and causing trouble. As you breathe air in, and it swirls around going down the tube, down the trachea, it's going to, the air will bump into the wall and it will smash into the wall and there's mucus along the wall. But that mucus is a little thicker, a little stickier. That mucus is like flypaper. You know how flypaper grabs the bugs you don't want around? It grabs the pollen, it grabs the bacteria. Everybody good so far? And when it does, then we have the cilia beating to move the mucus up the trachea to the back of the throat so you can swallow it down to the stomach where the acid looks at those bacteria and says, kills them all. Does that make sense? Okay. With sound effects, I promise. Okay, moving on. This making sense so far? All right, so one of the specializations you have is cilia, one of them you have is microvilli, one of them is goblet cells. Let's do one more. This is stratified squamous. We'll talk about in a minute. This is stratified squamous. Would you agree a scratch on the inside of your cheek is a lot worse than a scratch here? Why is a scratch on the outside not so bad? Because those cells are tougher. They're full of a protein called keratin. Keratin is the stuff that your fingernails are made out of, stuff your hair is made of. It's the stuff the horn of a rhinoceros is made out of. It's the stuff that cow horns are made out of. Stuff horses' hooves are made out of. Keratin is tough. Are you with me? So if we say that a cell is keratinized or has undergone keratinization, that means that it is full of a tough protein and it makes it hard to destroy that cell. Good? Now, an example of that we already talked about. I'm going back to your lab manual. And let's go back here. This top part of the skin, the stratum corneum, yeah, come on, focus. This even the thin stuff is about 20 cells thick. And all those cells, they're already dead. They're basically membrane sacs of keratin. They are tough. Does that make sense? Okay. So why is it that if I'm walking along and I bump the corner of something and I just scrape a few cells off, or I'm scratching, oh I itch, I'm scratching, I scratch off some skin flakes, I don't care. Because there's more underneath that and underneath that and underneath that and underneath that. Let me say it another way. Stratified tissues are really good at abrasion resistance. Good? So think of your mouth. You're chewing. When your food is rubbing on your cheeks, what would happen if that was simple squamous? You'd bleed after every meal, right? You'd be tearing holes in your cheeks and you'd have nothing that... So it's stratified squamous. Let's be gross. Let's talk about what happens when you go to the bathroom and you have to pass the feces out of your body. Well, would you agree that's rubbing on the wall of the rectum? Yeah? You don't want simple tissue there. You want layers. You want to be able to rub some cells off. Do we agree? All right. So that's stratified tissue. What about when you're eating and you swallow and the food runs down the esophagus to the stomach? It's rubbing along the stomach, along the esophagus the whole way. Do you agree? Stratified tissue. This, because of the keratin, is just the keratinization, is just super resistant stratified squamous. Does that make sense? All right, so we've gone through the three specializations. Do you agree? The stuff that's on tables 5.1, 5.2, 5.3. Let's now go back to the study guide. I'm not flipping over, but I'm looking right here. We said that objective 2 was know the function and location of the 4 epithelial tissues in table 1, yes? And we've done some of that, but let's go finish it up. So simple squamous, function, good at gas exchange, good at filtration, yes? Okay. Locations, glomerulus of kidney, location, lungs, location, lining of blood vessels and lining of the inside of the heart. Why? Super slippery. Lining of the organs as serous membranes. Why? Again, super slippery. Does that make sense? Are we good? All right, so we've done simple squamous, and you know what it looks like, right? You've seen the picture? Yeah? Okay. Guess what? On the test, or on the quiz, I might ask you, here's a picture. What is that? Oh, it's simple squamous. Or I might say, where is this located? Or I might say, what is the function of simple squamous? Something like that. I can ask it all different ways. Does that make sense? Are we good? All right. The next one is simple cuboidal. We've done that. We talked about that one. Kidney tubules are the main one, yes? Kidney tubules are where we do a lot of exchange. We do a lot of absorption and secretion. A lot of the kidney tubules, a lot of the cuboidal cells are doing that. They have some microvilli on top to make it easier to do the exchange. But you know that if you're looking at simple cuboidal, you're going to have one layer because simple. And you know that those nuclei are going to be round because that's the thing you can see for sure. The cell shape, the cube, depends on the way the slide was cut and you're looking. But the cell will have a round nucleus. Everyone good with that? Yep, okay. All right, the next one, pseudostratified columnar. And I'll just tell you right now, with the cilia, this is going to be the lining of the trachea. Good? Pseudostratified columnar, that'll be trachea for the location. Function, goblet cells to make the mucus, cilia to move the mucus away. So you can swallow it down and kill the bacteria. Are we good? Yep, not enough nod, are y'all with me? All right. And the fourth one that says no is stratified squamous. Abrasion resistance. Anywhere you got abrasion, you want to have layers, you can rub some off and not have a problem. Does that make sense? That's what stratified squamous is for. So where? Our skin, but also the inside of our esophagus, the inside of our rectum. I didn't say it before, but let's be honest, the inside of the female reproductive, well, not the whole thing, but the inside of the vagina, there is friction, there is abrasion, so you have lined with stratified squamous. Does this make sense? Everybody good? Okay, so would you agree we have done the 4 epithelial tissues that are listed in the study guide? Yep? All right. I'm going to do 1 or 2 more that aren't in the study guide. I'm going to use your lab book. I'm going to flip back to it. All right. So in your lab book, we're going to that table 5.3. And in 5.3, I want you to see. No. That's. Here we go. Yes. Transitional epithelium. Transitional epithelium can transition. It can change shape. So let's draw this because this is some strange stuff. Let me darken it up. That might be easier for you to see so you know I'm not just making this up out of my imagination. You see the outside of the cells better now? Yeah? Okay. Would you agree these cells are cuboidal-ish at the moment? But they've got really arched apical surface, the surface towards whatever is going on in there. That's the lumen of the bladder. That is the bladder when it doesn't have any urine in it. But when it fills up with urine, we have a visual aid. What I wanted you to say is it goes from thick to much thinner. Do you agree? All right. So these cells, these transitional epithelium cells, when you fill the bladder with urine, they get stretched and they get thinner. They go from being curved to almost looking squamous. They can change shape and snap back once you empty your bladder. Does that make sense? Everybody good? All right. That's it for epithelium. We got lots of other types, but those are the biggies I'm going to focus on because those are the ones that are in the study guide. All right, so I talked about epithelium. That was the first of the 4 major types of tissues. Good? All right. Anybody remember the second one that I said? Yeah, connective tissue. So what I want you to know for connective tissue, we're going to go through some examples. Oh, by the way, how will you practice this stuff? If we don't get through all that, what can help you prepare? Anybody look at what's happening before the end of the week in quizzes? Oh, I'm not going to flip back. Just trust me. On the 18th, you have to have the quizzes done for epithelial tissue, connective tissue, and integumentary stuff for this week. Does that make sense? Everybody good? All right. So let's talk about connective tissue, which is the second type of tissue. It has 3 things. All connective tissue has these 3 things. Are you with me? Not all in the same amount. And we'll give examples of differences, okay? So, all connective tissue has specialized fibers. All connective tissue has a ground substance. That's the stuff that is in between the fibers, in between the cells. Good, good. All right. And all connective tissue has specialized cells. Now, those specialized fibers, black. Okay, that's not working. We got to get another color. So, let's start with specialized fibers. So I said, oh, there are 3 types of specialized fiber. Oh, sorry, I said these are the 3 things: the fibers, the ground substance, the cells. But what I want you to know is that in fibers, specialized fibers, we've got lots of different kinds of specialized fibers. Most of the time, not all the time, there are 3 of those that we deal with. And I'm going to write this down. That's why it's switching color. Good. So 3 types. And the ones I'm going to go for are collagen fibers, reticular fibers, and we'll pull up a picture that makes this more clear. Okay, and then elastin fibers. So collagen fibers, reticular fibers, elastin fibers. But there are others. Good. Some types of connectivity have other ones. Now, these 2 are kind of special. The collagen fibers and the reticular fibers are made out of the same protein. I hate the way they named this. They're made out of a protein called collagen. So, collagen protein. But I'm sort of like, why did you do that? If the little fibers have a name that's not the same as the protein, why can't the big fibers have their own name too that's not the name of the protein? But both types of fibers, collagen fibers, reticular fibers, are made up of the collagen protein. Are we good? Elastin fibers, this one makes more sense, are made out of elastin. It's another protein, elastin protein. We only have 2 spots in the body that we have a bunch of that. Anybody want to make a guess?
Your ear.
Your ear is one, yeah. Here, let's do a quick demonstration here. Hey, I can bend it, snaps back, bend it, snaps back, bend it, snaps back. I can play with my dog's ears all day, right? Okay. You can take it, bend it, bend it, bend it. Look how it snaps back, doesn't matter. That's because we have the elastin protein. It stretches, snaps back. Great. The other place is the epiglottis. The glottis is the name for the opening to your trachea. We have like a little trap door that goes down every time you swallow to make sure things go into the esophagus behind, and then it snaps back when you're not swallowing. So every time you swallow, boing, 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 boing. Elastic fibers. Does that make sense? Okay, so that's good. But let's do these together. The specialized fibers and the ground substance together are things outside the cell, around the cell. So we have a fancy schmancy term for them. Together we're going to call those the extracellular, because it's outside the cell, matrix. Good? Those are the extracellular matrix. And some ground substances have one type of consistency and others have totally different. You would agree this is pretty rubbery, pretty springy. Yeah, okay. The ground substance for bone is a mineral called either osteoid or hydroxyapatite. People use both names. That's tough, like concrete. Do you agree? It's still a ground substance. The fibers are running through it. Good, all right. And so you have different. The weird one, and this is one I was thinking of when I was writing this down. Anybody know what the strangest connective tissue is? Like, you don't even think of it as a connective tissue, but it is. Blood. Blood is considered to be connective tissue. Now, the ground substance is easy. That's the plasma. That's blood plasma. Specialized cells, that's easy. Of course, we got our white blood cells, our red blood cells. We have our fragments of cells that we call platelets. Yeah, we got specialized cells. No big deal. Fibers in blood? But you only see them when you've got a cut that you're trying to repair. Does anybody remember what the word, the suffix on the end of a word in anatomy, ace, means? A-S-E, that we talked about before? If a word ends in ace, it's a?
Enzyme.
Yes, perfect. Good answer. Name?
Gabrielle.
Gabriella's my favorite right now. All right, so yes, if it ends in ace, it's an enzyme. I'm going to give you another one of those. If it ends in O-gen, like whatever it is, O-gen, that's the inactive form that could generate the active form if you need to. Okay? I'll give you an example. The digestive enzyme pepsin, when it's not in its active form, we call it pepsinogen. Does that make sense? The digestive enzyme trypsin, when it's not in its active form, we call it trypsinogen. Well, floating around in blood has fibrinogen. And as the name tells you, if you activate it, it becomes a fiber. This is a protein that in its normal day, it's floating around in the blood, all coiled up in a little ball shape. Does that make sense? Okay, floating around. But if it gets close to a place that there's a cut, like say this is a cut wall of a capillary, cut wall of an artery, whatever. You got a cut in a blood vessel, and you're going to bleed out there. These damaged cells here, they're releasing chemicals that will activate the fibrinogen, turn into fibrin, and it will go. And then, as the blood tries to wash it out, have you ever seen walking along a path and you see a stream or a river, and there are branches down, and behind all the leaves are piling up, all the trash is piling up, everything. All of a sudden, those fibrinogens, they're fibrin now, and you have all the things like the other blood cells packing into there, getting caught. That's the soft beginning of a clot. Now, some of those things are fragments of cells, pieces of cells that we call platelets. The thing they break off of the big, real full cell is called a megakaryocyte, but you'll learn that in the circulatory. Anyway, in AP2. But when you activate those platelets, they grow all these extensions too. So they start doing the same thing. But the difference is when they're covering the cut with all the little extensions, eventually they're going to pull them back in. And when they do, that's what squeezes the clot, pushes the fluid out, and starts to make it that denser, hard clot that we end up with. Does that make sense? But the whole thing starts by activating a special fiber. It's not a fiber until you need it, but there is fibrinogen. So yes, blood is still a connective tissue, has all three things. Does that make sense? We'll talk more about it later. We'll show more pictures later. I'm going to go ahead and hit the other two. Are you guys okay if I erase this? You know the O-gen thing? Yep. Okay. I'm going to give you the two other tissue types. Then I'm going to keep that out of the way so you can take a photo. Then we're going to erase it. We're start going for pretty pictures. Good? All right. So the next one, number 3, we've got muscle. And you already know this, but we've got 3 types of muscle. In the heart, we have cardiac muscle. And that is involuntary. You cannot control that. All right? We also have, in other places, we have involuntary stuff, like say in the lining of your digestive system, the lining of blood vessels, we have smooth muscle. And that would be, let's call it tubular organs. Like I said, the gastrointestinal tube, the blood vessels, are we good? Yeah? Okay. And those, you don't think about to make it happen, but it does happen. Suppose you're walking around outside and you hear rustling. I heard stories that there was a panther in the area. All of a sudden, the blood going to your eyes, those blood vessels get bigger, so more blood can be delivered. We call that vasodilating. Oh, and guess what? The blood going to your stomach, those blood vessels will vasoconstrict. Because it's more important to run away from the threat than it is to, oh, I'll rest and digest. Does that make sense? Yeah. Okay. So, smooth muscles and tubular organs like blood vessels, like the digestive system. And then the third one, this is the main one, this is one we talk about so much, skeletal muscle. And if you look I am going to flip over. I know I said I hate flipping over because it takes so long. You're now about to see it. They'll eventually catch up. I promise. Don't make a liar of me. Okay, histology. Here we go. So, what's I going to do? I'm like ADDing. Skeletal muscle, brain and neutral. Okay, so we're looking at the thing. Here we go. What I wanted you to know, what we're going to do on this study guide, we're going to go over 4 different epithelial tissue types. Those are the main ones I'm going to worry about because those are the ones in the study guide. Those are the ones you're going to see on the quiz because those are the ones in the study guide. Does that make sense? Like, they have transitional epithelium. I might talk about it. I may put it on a full lab test, but I'm probably not because these are the 4 they're really hitting on for epithelium. These are the ones, let's see, 1, 2, 3, 4, 5, 6. These are the 5 that they're really hitting on for connective tissues. There are others, but in muscle they have all 3. So they want to know where they are and what they do. We've already talked about that. I didn't write it all on the board, but you agree we talked about all of it. The one cardiac muscle is in the heart only, it's for pumping blood, yeah? All right, so we've got those, but we'll get those. And then this bottom here, neuron. Oh, I remember what I was doing. I was coming over here because I was going to show you that to help you prepare for quizzes and tests, sorry, the little on-paper quiz and for the test, we have a practice quiz for the skin stuff, for the integumentary, the stuff that Monday and Tuesday mostly focused on. And you and I right now, we're focusing on the tissues. We're doing histology. So you agree we have a quiz here, a practice quiz on connective tissue. We have a practice quiz on epithelial tissue. And we have one on muscle and nerve. But look at the dates. Oh wow, I didn't. Okay, I thought I made this one different, the muscle and nerve. It's not. I'm going to extend that one because I'm probably not going to put muscle and nerve on the quiz next week. I'm probably going to review the muscle and nerve in more depth whenever we do muscle and nerve because they have their own chapters. They're not like the others, you know, where we do a quick overview and then get into the stuff. Muscle, we got multiple chapters on muscle. We got multiple chapters on nerve. We can fit that stuff in there, so I'm not too worried about it. Expect this one to change, but these 3 right here, they're due on the 18th. Get them. Make sure you don't get late fees. How do we not get late fees?
Before the due date.
Use every attempt before the due date. Every attempt. If they're 20, 25, use them all. Then you're not tempted to use them again and get late fees. What do you do if you want more attempts closer to the test to use them for practice? Yeah, you talk to me and say, I would like more attempts. Can you move the due date for me so I don't get a late fee? And I'll move it and I'll give you another 5 attempts or something. I'll give you more attempts. Does that make sense? That way you don't get hit with late fees. If you just try to do this on your own, like, I'm going to save 3 and then I'll use them later, you will get late fees. And late fees, it's in the syllabus, they're 5% a day. Well, I don't know if I put that in the syllabus, but they're 5% a day. I can show you the 5% a day. No, I can't because then I'd have to pull up the grade book and show you everybody's grades too. But if I'm in the grade book and I click on the upper right corner where they have like the settings thing that pops up, I can choose the amount, and late fees are 5% a day. That's for all my classes. That's generous and kind. Last semester they were 10% a day. So, anyway, you get the idea. So the reason I jumped back here was to show you, do not forget that before the end of the week you need to take the Histology Lab quiz, you need to take the connective tissue and the epithelial one. Are we good? Is that clear? Okay, I'm going to write it up here on the board so it's very clear. Okay. Quizzes. Oh. There, the dyslexia kicks in. You see it? D's, B's, and G's. I flip them. And P's. Yeah. So that was supposed to be integ. All right. Our connective and epithelial. These the 18th. Everybody good? Yeah, so take them, use them, know it. Okay, so but we've talked about the 3 types of muscle. We talked about where they're located, yes? We've talked about what they do. I'll say this one again more clearly. I said those are the muscles we usually think about. Those are the ones that when they shorten, they pull on bone and cause us to move, cause us to stand, all the things we normally think of muscle for. Good? All right. And again, you'll get a lot more of that as we go on. So the 4th type of tissue is nervous tissue. And I'll be more clear, there are 2 different types of cells in nervous tissue. Actually, there are a bunch of types, but we have neurons, I'll call that A, neurons. And then we have a bunch of support cells for the neurons we call glial cells, which is a horrible name because glial means glue. Back before we knew all the awesome things that they did, we were like, oh, they glue the neurons together, so we call them glial cells. But there are like 6 or 7 different types of glial cells. Are you with me? And when we get to the nervous system, you'll have to know them all. Right now, that's not the case. But I will talk about one other thing. Your textbook, all the textbooks, when they talk about neurons, let's see right here. I'm flipping back. We have 3 pictures of neurons. 3 pictures of neurons. All right, so the lab book exists. The lab book has pictures of neurons, and I'm going to flip back to it. Then I'll try to move this. Come on. All right. They have this drawing of a neuron, the classic drawing that's in all the textbooks. I mean, that's a photograph for one that really does look that way. And here they show them with all the glial cells around them. Yeah, that's great. What I want you to get, and I'll say it right now because I hate the way people do things. In anatomy, they'll be like, this is the neuron! And then you get to the actual chapter on the nervous system and say, but wait, the thing we told you, that's only part, there's more! So they're going to tell you, this is the shape of a neuron. That's the shape of one of the 3 main types of neurons, okay? Let's go. So the main shape of a neuron, you have a cell body that's kind of spiky, has pieces coming off they call dendrites. They're like branching things. Usually, not always, those are considered to be the input part of the cell. Then we have a long axon that goes off, and then we have at the end the axon terminals. Okay, great. That's what most drawings show, and that is one of the types of neurons that does exist. But that is a neuron that we would call a multipolar neuron. Multi, because many directions. We've got branches, poles going off every way, and one really big one there. Do you agree? We also have, and none of this matters right now for the upcoming quiz. None of this will matter until the nervous system test, okay? But we also have, coming up, when we get there, cell body, and it's like it's been moved to the middle of that axon. They actually call this whole thing dendrite and that axon to avoid confusing it. But this, would you agree we've got 2 main poles off that nerve? That's a bipolar neuron. Does that make sense? Not enough nods. Y'all with me? Okay. So multipolar neuron, I'll put out polar neuron, and then we have a bipolar neuron. And then the truly weird one, we have neurons that have a cell body that looks just like a normal cell, and then it has one little extension, and it goes to the axon. You know, like our nucleus is over here, and we have our dendrites here doing our thing, and then we have our axon terminals over here. And this, because there's only one piece coming off of the cell body, we would call this a unipolar neuron. You with me? Now here's the really weird part. Textbooks, until you get closer to the nervous system, they act like they're all like this. All your sensory ones are like these 2. I think they're pretty important. I would have been mentioning them from the start, but I didn't write the textbook. Are we good so far? Okay. So you know the glial and support cells, you got like 6 of them. You know you got 3 different shapes of neurons. You know that neurons and glial cells are the parts of the nervous system. Are we good? Yep. All right, we have done the 4 types, and we've done it in way more detail. We're not just rattling off the names, but objective 1, name the 4 primary tissue types found in the body. We got that. Do you agree? What I'm going to do now, turn this around, try to make that less visible. All right, I'm going to say, everybody take a photo. I'm going to erase. Everybody take a photo. I'll hold it so you can use your phone to take a picture. Okay, yeah, fine. So everybody take a photo real quick, then I can erase all the things on the board. Okay? Y'all good? All right, didn't see that many phones go up, but we're good. All right, I'm now erasing. Nervous system be gone, muscle system be gone. Connective tissue be gone. All right, we got that. Epithelial. So we've done good work so far. We've covered a lot of details. Let's now hit the next objective. The next objective, note the function location of the 4 epithelial tissues listed in Table 1. So I'm going to scroll down to Table 1, and it wants you to know about simple squamous, simple cuboidal, pseudostratified columnar, and stratified squamous. There are other epithelial tissues, but those are the ones in Table 1. Yes, you guys see that? Okay. We're going to talk about them, but before we do, we're just going to do a real quick review. All right, I'm going to draw a tissue, and you tell me what it is. We got a basement membrane here below the tissue. These are our cells. What kind of cells would these be? Oh, yeah, but the full name of the tissue would be simple cuboidal, because it's 1 layer. Does that make sense? So, yeah, cuboidal would have been a half credit answer. Simple cuboidal would have been the full credit answer. Good? All right, now let's modify. What kind of tissue do we have? Remember, tissue is both the layering and the shape of the cells. Louder? Yeah, you got simple columnar, exactly. All right, now, now what do we have? Stratified columnar. Everybody good with this so far? Now we're going to go for the weird one. This is not one of those 3 simple shapes that we've been talking about. This is one that messes a lot of people up if they're not paying attention. So, I'm going to draw a tissue. It's one that a lot of people think has layers and it doesn't. Cells have weird shapes. We'll do a few of them here the easy way. We'll make some that are a little more realistic and strange shapes. Imagine you have a mixture of cells. It might be like, think of them as wine goblets and upside down wine goblets for the basic shape, like that. Then you got a couple weird things that they don't even go to the top, not even up to the apex. We've got this going on. Anytime you have a cell with a big, fat, thick area, that's probably where the nucleus is going to hang. All right. Now, you look at this. Suppose you were lousy with a microscope and you didn't know how to focus. You couldn't get the membranes visible very well. You'd see row of nuclei. You said, that's stratified. It's not. How do you know it's not? They're all attached to the same basement membrane, all attached to that same collagen fiber at the bottom of the tissue, yes? Even ones that don't go all the way up to the apex. These are all one tissue because they're all attached to 1 layer at the bottom. Good? So you know it's simple. But because it looks stratified, we have a special name for it. We're going to call it pseudostratified. In fact, because they're tall, you could like maybe be pseudostratified columnar. In fact, let's change one of these because it looks so much like it. Let's say this is light staining. I probably should have used black. That has a bunch of granules in it. This would be one of those single-celled glands. Give me a G name. Goblet cells. Perfect. And they make what? Give me an M. Mucus. Yeah, they make mucus. They make mucus. If you had this, let's add one more detail. We're getting a specialization. And I know with my drawing, it's hard to tell the difference. So I'm going to draw the feature, and then if you tell me, I'll tell you it's one of the two, and you tell me which it is. So we got this thing going on the top. You think we're dealing with microvilli or cilia? Well, actually, this one's supposed to be cilia, but my drawings are rubbish, so that's fine. But this, goblet cell laying out mucus, all those pseudostratified cells with the cilia, that's the lining of your trachea. Does that make sense? Oh, so let's say it another way. Hey! Pseudostratified columnar, item 3 on table 5.1. At the back of your study guide, you need to know that. What is the function? It moves the mucus with all the nasty things trapped so it doesn't get into your lungs. What is the location? Lining of the trachea. Everybody good? All right, good. So we've got that. We've done one of those. Let's now do the others. All right. Let's try using your book, your lab book. If not, we'll very quickly move to the lecture book where everything looks better.
I'm sorry, what was the function of the simple cuboidal?
I was just about to do that one. We haven't done simple cuboidal. Simple cuboidal we did. I thought you, for when I was thinking simple squamous. Simple cuboidal is absorption and secretion. And it said that in your lab book on...
The one that you just drew, wasn't that
page 95. That was pseudostratified columnar.
Okay. What was the function of that one?
It's protected because it moves the mucus. Okay. Here, I'll take your lab book and put it right there. Pseudostratified columnar epithelium, protection, secretion, and movement of particles when the epithelium is ciliated, as it is with all the cilia sticking up in the lining of your trachea. Good? Right. So location, trachea, function, protection, secretion, movement of particles. Are we good?
Yeah, where was the location of the simple cuboidal?
Oh, okay, thank you. People are pulling me forward and back. I was trying to say that, but I pointed out before, I said, oh, this is a nice picture right here. This is simple cuboidal. I said, that is a tubule cut longwise. Anybody remember me saying that? Anybody? Nobody. I promise I said it because I'm going to then show you simple cuboidal when you cut across the tube. The wall of the tube is made out of simple cuboidal. That's a kidney tubule. Good? Good. All right. And if you look, simple cuboidal is one of those things you can mess up very easily because it's not, more than any of the other tissues, this is a use your imagination when we tell you it's cuboidal kind of tissue. Okay? Let me explain. There are some that very much, I'm tracing an outline here, resemble a cube. And they have a nice big round nucleus. But there are some that's very much imagination to call that a cube. So what do we know? If it's kind of the same height and width and the nucleus is round, we're going to call it cuboidal. Even if it clearly is a trapezoid shape, because we don't have trapezoidal tissue. Does that make sense? That's still simple cuboidal, even though it's not looking very cube-shaped. This one's even worse. Look at that. Y'all with me? Yeah? But that's simple cuboidal. And this, again, is still kidney tubule. See, it says kidney tubule. The other one, I don't know if it said or not, but it's still kidney tubule. Good? Yep. So a location for simple cuboidal would be kidney tubules. There are other places, but anywhere you need to do a lot of secretion and absorption. It's good at absorbing things. In fact, when we drew it on a PowerPoint, we had it looking like the lengthwise cut, but the thing that helps it to be good at absorption a lot of times, of course my marker fails at the moment, it has a lot of microvilli to increase the surface area at the apical surface, so it can absorb things. Does that make sense? Are we good? I'm not getting nods, are you with me? All right, good, good, good. But simple cuboidal, good at secretion and absorption. All right, now simple squamous. Simple means it has how many layers? One, exactly. So let's see if they have a good picture here. Yeah, okay, so your lab book has rubbish pictures. We're going to go for your textbook instead. No, these are actually pretty good. All right. So, here we go. Yeah, this is a good picture. Right. I'll move the camera instead of moving the book and having the book fall off. This is lung. This is the air sacs in the lungs. Let's talk about that. Here, you have capillary. And here you have air. Look at how thin the barrier between air and blood is. It's 2 cells thick. One flat, simple squamous cell makes the wall of the alveoli, the air sacs, and another one makes the wall of the capillary. So you have 2 right there. If you're only 2 very tiny flat cells wide, it's easy for oxygen to go from the air to the blood, and for blood to send the carbon dioxide back to the air so we can breathe it out. Does that make sense, y'all? Okay. So where do we have simple squamous? Anywhere we have to do filtration and absorption, but it's really good at filtration. Air in the lungs, but also the glomerulus, the filtration membrane of the kidney. I'm going to read the way they describe it, and then we'll move on. So location, air sacs in the lungs. Oh, I forgot to say it, lining in the blood vessels. Well, I did say it. The capillary wall is only 1 cell thick. Remember I said that? Here's the deal, though. You'll get this when you get to circulatory, when you get to AP2. Most blood vessels have 3 layers, okay? The inner lining, which is super slippery smooth, it's simple squamous. And we used to call that the endothelium or the endometrium. Good? Then, in the middle, we have the myometrium. That is the thick layer that has the muscles that do the vasoconstriction, vasodilation, yeah? And then outside that, we have the tunica externa, which is the outside, is thicker. Okay, full connective tissue, the whole thing together. Capillaries are so thin to be good at gas exchange, they have lost the other 2 layers. They don't even have them. They're just that smooth inner lining. Does that make sense? And that lining, it's not just the blood vessels, it's also the lymph vessels. The serous membranes that we talked about before, the one around the heart, remember the pericardium, and the one around the lungs, the pleura, those are made out of serous membranes. So they're very thin and they just have fluid they trap for lubrication. Does this make sense? Are we good? All right. So that's your location. And in case you can't see it very well back there, I could change the contrast. But there's the nucleus. It's very squashed flat. Just like in the columnar, the nucleus is squished vertically. Does that make sense? Are we good? All right. So this is simple squamous. Tell me how good you think your lungs are. What would happen if you inhaled a bug and it got all the way down your lungs and it's scratching the lining of the lungs, the alveoli? Do you think they'd survive that very well? No. Because it's very, very weak. It's only 1 layer thick. You rip that layer, and now you're a capillary and blood, and oh, big mess. What do you use if you want lots of protection? You want lots of layers, am I right? Okay, so stratified squamous is the one we use for lots, and that's number 4 on that table, the table 5.1 that we're talking about. Stratified squamous, this right here is stratified squamous, the lining on the inside of my cheek. Why? Because if you're chewing on food, it's scraping the inside of your mouth. If it was simple, you'd start bleeding as you scrape it. Does that make sense? So you're eating potato chips and you get a little scrape. Ah! Well, we don't want that. So we have layer after layer after layer after layer. Good. This is also stratified squamous. It's just that this is keratinized stratified squamous, so it's tougher than the non-keratinized inside your cheek. Are we good? Does it make sense? But anywhere in your body you need abrasion protection, you're going to go with stratified squamous because, like the skin, okay? Skin, the outer layer, the stratum corneum, it can have like 20 cells thick. So if I scrape 1 or 2 of them, if I run into a desk or something, man, who cares? Because I've got a layer under that and under that and under that and under that and under that. And that's just the outer part of the skin, the stratum corneum, the outer part of the epidermis. Does that make sense? Okay. The next chapter they talk about how the epidermis is the stratum basale, and then the stratum spinosum, and the stratum granulosum, and then stratum lucidium, if you have it, that's only a thick skin thing, and then the stratum corneum. And the corneum by itself is 20 cells thick usually. So that's the power of stratified. You scrape some of those off, it doesn't matter. You've got so much underneath to protect. Does that make sense? So that protects your cheek, but let's get gross and go the other end. Say you have to go to the bathroom and you're passing poop, passing feces out of your body. What do you think the rectum is like? It's going to be stratified squamous, right? Right? Because abrasion resistance. In fact, there it's more important. See all the nasty bacteria with it too. Yeah. Yes.
So is the stratified squamous, is that the most abundant and?
That's a really good question. I don't know. It's certainly the most abundant in places you have abrasion. So the lining of the lining of the rectum, the lining of the mouth, lining of the esophagus, the outside skin on your body, up the lining of the female reproductive tract. Of course, you got a lot of it there. Overall, I don't know, because you really don't have that in places you don't have rubbing. So, say between the gastrointestinal tract and the body wall here, I don't think you'd have much of that. Does that make sense?
Yeah, just because I figured that everything, like it's your body, you need protection.
Well, there are some things we do, but we just talked about how the serous membranes are simple, so stratified. And remember, so like around the intestines, around the liver, that was peritoneum, that's serous membrane, that's going to be simple squamous.
Okay.
So, but I will say there's a heck of a lot of it. It was a good hypothesis. Does that make sense? Are we good? Yeah. All right, so would you agree we've talked about stratified and where it is? We've talked about pseudostratified and where it is. We've talked about simple cuboidal and where it is, yes? We've talked about simple squamous and where it is. Hey, I think we've taken care of the epithelial stuff. Let's go back and look at that objective and see. Yes, there are other epithelial tissues, but those are the ones that are on that study guide. Now, if there are more on the practice quiz, I might put the things in the practice quiz on the paper quiz that'll be next week, or on the test. Basically, I wrote those quizzes, the practice quizzes, to help prepare you for what I'm going to be doing in tests. Does that make sense? Anyway, so know the function looks to the 4 epithelial tissues listed in table 1. I think we did that. Good? Next objective. Know the functional location of these 6 connective tissues listed in table 2. So I'm going to jump down to look at the 6 connective tissues. And the ones that has are areolar, adipose, reticular, hyaline, osseous, that means bone, and blood. So let's talk about them. When you see areolar, think open to the air. I know it's not spelled the same as AIR, but what I'm getting at is that areolar tissues have lots of space in them, Okay, we're in connective tissue, so before we do that, I'm going to talk more about connective tissues. I said I would tell you more about it. What were the 3 types of things in all connective tissues? Specialized what? Fibers? Yep, perfect. Specialized cells, yes? Well, all of the connective tissues have all of those 3 things, but not in the same amount. Some of them, like bone, lots of ground substance. That's what it's famous for. But it has fibers too. You just don't usually see them. Some of them, and I'm going to go for pictures right now to show you some, then we'll move on. Okay? So, if we're looking here, that's mesenchyme. We don't care about that. We're not testing on that. Okay, so connective tissue proper. Here we're going to have areolar tissue. This is in your textbook because they're better pictures than your lab. This is in your textbook on page 169. I would recommend writing that down in your notes so it's easy to get to. But this is areolar connective tissue, and it looks very open, like there's nothing there. Like you've got fibers for sure, and you've got specialized cells living there. This is not nothing. This is not air. This is ground substance. There are things there. Does that make sense? But it's got a lot of space. And when you look at this areolar connective tissue, it has elastic fibers, it has the bigger, thicker collagen fibers. See how here we have the elastic fibers are thin, little stretchy things, and then the thicker ones, those are the collagen fibers. Everybody good with that? Okay. This is areolar connective tissue, and let's just do what they say right here, talking about it. Structure. Scattered fibroblast. Oh, we're going to introduce a new suffix. Everybody ready for this one? When you see blast, I'm going to give you 2 words that I want you to think of. Okay? When you see something blast, the B should remind you of baby, because that cell might grow up to be something else. Okay? But it should also remind you of builder. Because there are no child labor laws in anatomy. We put the baby to work. Does that make sense? So a fibroblast, you probably guessed, builds fibers before it grows up into something else. Does that make sense? Okay. Guess what? In bone we have an osteoblast. What do you think it does? It builds bone matrix. Good. In cartilage we have a chondroblast. What do you think it does? It builds cartilage. Are you with me? Is it making sense? Okay. But blasts are baby ones. And you can see we've got, both in the drawing and the picture, we got a whole bunch of those specialized cells, right? And what are they going to be doing? They're going to be making substances. Where it says here that the areolar connective tissue has abundant viscous ground substance, think of it as being like a jelly, okay? When we talk about viscosity, we're usually talking about things like how thick is the oil in your car. I know I'm not supposed to talk about cars. Well, if it's really thick fluid, like a jelly, does that make sense? So it's got some give to it. It's not like solid like bone. So ground substance doesn't have to be solid. But you already knew that because what's the most fluidy of the connective tissues? Blood. Yeah? So blood plasma. Anyway, but it's viscous. It has a few elastic fibers and collagen fibers, a lot of blood vessels. Okay, great. This is what it looks like. Protects tissues and organs, binds skin and epithelia to deeper tissue, space for blood vessels and nerves. Since there's a lot of blood vessels in there, anywhere we need to pass blood vessels from one area of the body to another, we might have some areolar connective tissue around it, okay? Protects tissues and organs? Yeah, well, if you got all that gooey, squishy kind of ground substance, if you surround things that you don't want to have smacked into, there's a little give, a little cushion. Does this make sense? All right, so location, the papillary layer of the dermis. All right, now, this is something that is in the next, and we're going to use the good book. I know we'd use your lab book, because this picture is pretty good. Remember, this is the thing I was talking about with the other class earlier this week. So let me jump to skin. Here we go. Oh, heck! Okay. We could use this picture. But we can get bigger because we have the same models. We're going to use the model, okay? So I'll just say this really fast. They said the papillary layer. Okay, so when we are looking at the skin, everything on this purple and above, this is epidermis. Everything below it, down to the yellow here, this is dermis, this is epidermis, this is hypodermis, below the dermis. Are we good so far? Okay. So, when we look at that, I already told you that the top, the epidermis, has layers that we call the stratum corneum. That's the one on the outside that's in pink. And below that, the ones in purple will be the stratum, well, this, because it's thick skin, has the lucidium. Lucidium is only found in thick skin. The stratum corneum, lucidium, and then here we have granulosum, spinosum, and at the very bottom, at the base of the epithelium, we have the stratum basale. So those are the layers. And the mnemonic to remember is come, let's get sunburned. In order from the outside where the sun is to the inside. Does that make sense? Come, let's get sunburned. Is stratum corneum, lucidium, basale, spinosum. That's why I said that. Granulosum, spinosum, basale. Good? Yes? All right, good, good. So there you got that. But here, do you see, let me get a little pointer. Here we got a little sticky uppy part of the dermis, a little sticky uppy part of the dermis, a little sticky uppy part of the dermis, a little sticky uppy part of the dermis. Those things that are sticking up, the anatomists who saw them, they thought they looked like fingers sticking up, so they called them dermal papilla. And the top part of the dermis is the papillary layer. Good? Good? Yep. Anything below that is full of reticular fibers, and that's the reticular layer. Are we good? And that's like 80% of the skin. Let's pull up your lab book again and show the picture. All right. So you can see the papillary layer is this little tiny part right up here where the dermal papilla are. Good? But most of the, okay, that's not making me super happy. I'm going to go for the other book. But most of that is the reticular layer down here. Let's go for the other book that has a prettier picture, as it usually does. All right, so here, yeah, this is a much prettier picture. Check this out. So the papillary layer is that little part at the top of the dermis where the dermal papilla is, yes? And then most of the dermis is the reticular layer. Are we clear? All right, now let's go back to what we were just looking at in histology and what they said. Right there. Where is it? Areolar connective tissue? In the papillary layer of the dermis, the subcutaneous layer below the deep to the skin, we're talking about down that hypodermis area, okay? Around organs, nerve cells, some muscle cells, we have some of this around them just to cushion them, not have things harass and get to them. Does that make sense? All right, so we've got the first of the ones on there, areolar connective tissue. The next one is the adipose tissue, and that's the next picture. This is a perfect picture because it's a drawing. This is a photograph, so it's more accurate, but it's hard to understand what you're looking at. Let me make this a little darker so it's a little easier. When you look at all the white, no, do not do that to me. When you look at all the white things here, you might be thinking to yourself, oh, well we said all the white stuff here was the ground substance. That must be the ground substance. No! There's almost no ground substance. Remember, I said that some tissues have more of one, more of the other. Adipose tissue is almost all cells. These cells, I'm going to make it lighter now so you can see the picture again. Look at these specialized cells. All of the nucleus and cytoplasm is shoved off to the side. There's a big lipid storage vacuole here. That is lipid storage. The reason it looks white, do oil and water mix? No? Okay. Well, guess what? Most of the stains we use in anatomy are water-based. So since they're dissolved in water, if you cut a piece of tissue and you put it down a microscope slide and some of it's fatty, and then you put the stain on it, you rinse off, none of it stuck. None of the stain stuck because it was dissolved in water, and water doesn't like fat. So it looks white on the slide. It's not that there's nothing there, it's full of fat. Does that make sense? Okay? So, but what I'm getting at is these are all three connective tissues. And you can see when you're looking at the areolar, a lot of space and a lot of fibers. Are you with me? You have cells, but they're not the main thing. Now you look here, you look at the adipose tissue, and you've got all three things, because you always do. But it's mostly cells. There is space, but there's not much ground substance, and you'll be there's more there. And you'll be really hard pressed to find the fibers. You have to look a long time on a slide to come across a fiber, because there's not much room. Does that make sense? Everybody see it? Okay. Now this is reticular fiber. I told you that there were three types of fibers. You may remember the three types of fibers. I said three main types of fibers. Collagen, reticular, and elastin, yes? Okay? So this has a lot of reticular fibers. Those are made out of collagen, but they're really fine, diffuse, spread out. They're the scaffolding that these white blood cells are crawling around on, hanging out on. This picture tells us this is the spleen, but it could have been a lymph node too, part of a lymph node. But so you have these reticular fibers, you have this space in between, you have these white blood cells hanging out here. You know what's going on there? It's part of the lymphatic system. You got all the lymph going through all that open space, and the white blood cells are sitting there just watching. I know how to explain it. This is an old movie. It used to be like, I would use this because this is a new movie, but I'm so old now that it's an old movie most of you guys have never seen. Probably it came out before most of y'all were born. Anybody ever see Will Smith in Independence Day? Okay. All right. Well, here's the short version. He flies a UFO that was at Area 51 up to fight the aliens in space. The end. Okay? That's the whole plot, whatever. And there's a spot where he has to fly the ship they stole from the aliens up into the alien mothership. You with me? That alien mothership is full of steel girders in every direction with little fighter UFOs hanging off of them, watching to see if something suspicious goes by. And of course, once they spot him, they all come off of it and chase him through the mothership, you know. So, but that idea, all these white blood cells are hanging out on the reticular fibers, looking for bacteria and bad things coming through.